Hello boys, welcome back to another session of video tutorials. And in this session we are going to discuss about the cell cycle, various stages or phases that happen in cell cycle. And this session is meant for class 11th biology students, and this is for 30th July 2020. Our memory helps us in tracing out and reflecting upon what we have been discussing in our previous sessions. In our previous session, we were talking about the significance of the cell cycle, how many stages or phases will be there in a cell cycle, what is a cell cycle, etc. So the sequence of events by which a cell duplicates its genome, synthesizes cell content, and eventually divides into two daughter cells is termed as cell cycle. That's how we have defined cell cycle. So a typical eukaryotic somatic cell has two main stages. The long undividing interphase, that's called I phase, and a short nucleus dividing stage called M phase. So there is a I phase and M phase. And this M phase is followed by cytokinesis. Cyto means cell, kinesis means breaking of the cell, cytokinesis. Whereas in M phase, it is the nucleus that is breaking. So we say karyokinesis, carry on nucleus, kinesis breaking. So first breaking of the nucleus, and then that is followed by breaking of the cell. Karyokinesis is followed by cytokinesis. And the interphase is metabolically active stage. As I said, it was wrongly interpreted previously in olden days that interphase is a resting phase. But the latest research shows that it is not the resting phase, rather it is preparatory phase for the possible division of the cell. Hence, interphase is a very metabolically active stage. And it is divided into three phases as we discussed, leaving out the G0 phase. So interphase mainly consists of G1 phase, that is growth 1 phase, S phase, synthetic phase, G2 phase, growth 2 phase. So interphase has got three subphases: G1 phase, S phase, and G2 phase. And G1 phase is a stage of initial growth. As we are seeing here, G1 stage is the first growth stage, initial growth stage after cell division. So cells mature by making more cytoplasm as well as organelles. And cell carries on its normal metabolic activities. So these are the main features that take place in G1 stage. As I said, G1 stage is the initial growth of a newly formed cell in which the cell carries on active metabolic activities in preparation for DNA replication. In G1 phase, a cell has three options: to continue cell cycle and enter S phase, stop cell cycle and enter G0 phase, that is back phase, for undergoing differentiation, and get arrested in G1 phase where it may enter G0 phase or re-enter cell cycle. Some cells such as nerve cells, neurons, and erythrocytes, RBC, that do not divide further exit G1 phase to enter an inactive stage called G0 or quiescent stage of the cell cycle. They remain metabolically active but no longer proliferate unless they are called to do so. Next, we come to S phase, that is synthetic phase. It is the synthesis, preparation of DNA. Either the new DNA is prepared, it is copied, or it is replicated. DNA replication takes place. That's why we say S for synthesis. Synthesis phase marks the period during which DNA synthesizes or replication takes place. During this time, the amount of DNA per cell doubles. As you are seeing, the DNA amount is getting doubled. If the initial amount of DNA is denoted as 2C, then it increases to 4C getting doubled up. 2C has become 4C. However, there is no increase in the chromosome number. Remember, chromosome number remains constant. It cannot change, because if the chromosome number changes, then the cell will lose its identity. So chromosome number will not change, but the content of the DNA gets doubled up. How does it get doubled up? Because of DNA replication. So DNA gets replicated, and DNA replication begins in the nucleus, and the centriole duplicates in cytoplasm. So during this S phase, most important one is DNA replication. And the next, the cell enters into second growth stage, G2 stage. It is a stage of further growth of the cell and its preparation for division. During this stage, synthesis of RNA, ribulose, nucleic acids, and proteins take place. So it occurs after DNA has been copied or replicated. All cell structures needed for division are made, like centrioles, very important ones. Both organelles as well as proteins are synthesized. These are the important events that take place in G2 phase. And what's happening in this, all the three stages together? So what the cell looks like, it's, as you are saying, will be looking like that. And within that, there is a nucleus, the nucleolus, and centrioles which are replicated, and then astral bodies, as you are seeing. And the nucleus has got nuclear membrane, and nucleus is very prominent, so this is eukaryotic animal cell. And the chromatin gets replicated. And what is the important event that takes place? A DNA replication, centrioles also get replicated, and then cell prepares for possible division. So broadly speaking, we can sketch the cell cycle in this fashion. That is, I phase, interphase. Now you can see interphase has got three substages: G1 phase, S phase, G2 phase. And M phase has got, broadly we can say, 4 phases. They are called PMAT. P for prophase, M for metaphase, A for anaphase, T for telophase. Pro means primitive, first. Phase means stage. So it is the first stage, prophase. Meta means middle, phase, stage. It is a middle stage during cell division. Ana means dividing. It is almost the chromosomes are moving to opposite poles, anaphase. And telos means breaking. What is breaking? Telophase. Nuclear division takes place, and then this nuclear division is followed by cytokinesis. Cyto, just now I said cell. Kinesis means breaking. The cell is getting broken. So overall, if you draw the cell cycle, it looks like this. You got broadly 2 phases. One is interphase, another is mitotic phase. I phase, M phase. In I phase, there are 3 substages: G1, S, G2. And in M phase, you got 4: PMAT, prophase, metaphase, anaphase, telophase. And this will be followed by breaking of the cell, cytokinesis. So single cell, by undergoing the cell cycle, forms 2 equivalent daughter cells. So the significance of this is that it forms, as I said, new cells. So old cells will be giving away to the formation of new young dynamic cells. As a result of that, the tissue grows. Tissue will be formed and tissue growth takes place. As a result of growth of tissue, the organs also will grow and organ systems will form. Organism as a whole will grow and develop. So thanks to mitosis, the cell cycle, the leading to formation of new daughter cells. And second, worn out cells. The old cells will be removed. In the place of old cells, new cells will be formed. That keeps the organism dynamic. And repair, if there is any cell damaged, the damaged cell through cell cycle can be repaired. Hence, it occurs in every part of the body of an organism. There is no part where mitosis or cell cycle will not take place. We will be studying in our next session in detail what are the events that take place in prophase, metaphase, anaphase, telophase, and finally how one parental cell gives rise to two equivalent daughter cells. So please draw the diagram, write the important events that take place in G1, G0, S phase,